Hello students, welcome to today's session. In today's session, we are going to discuss about the methodologies or identification of human DNA. So let us see the human DNA. So this human DNA has two parts. One is called as coding part, the other one is called as the non-coding part. So the coding part codes for the protein in transcription and translation of the proteins. And the non-coding part is heterochromatinized and this will not code for anything or it does not give anything. So let us identify the human DNA. So to identify human DNA, there are two approaches. The first approach is to sequence only the coding part, that is known as express sequence tags, that is EST. The second approach is to sequence entire DNA, that is coding part or non-coding part. That's why it is called as blind approach. It is also known as sequence annotations. And before sequencing of a DNA, the cloning of DNA is required. And the process for cloning a DNA is, the first process is, firstly we have to isolate the total DNA. This isolated DNA is made into fragments by using restriction endonuclease, and this restriction endonuclease is called as molecular scissor as it makes the fragments of the DNA. These DNA fragments are introduced into the host through the vectors called as, one is BAC, that is bacterial artificial chromosome, and YAC, that is yeast artificial chromosome. So through this vector, these fragments are introduced into the host where the cloning of DNA takes place, or DNA are ready to clone. And for entire process or sequencing of DNA, there are two methods. One is used as shotgun method, the other one is Frederick Sanger method. So by this process, the DNA sequencing is done. The next, let us see the salient features of Human Genome Project. The Human Genome Project contains 3164.7 million nucleotide bases, and the average gene consists of 3000 bases. The largest human genome that codes for the protein is dystrophin. That means there are 22.4 million base pairs, and the total number of genes estimated till now are 30,000. The functions are known for only the 50% of the genes still discovered. That means the out of the total discovered genes, only the 50% function is known. Less than 2% of the genes code for the protein. So in our body, there are 23 chromosomes where out of those 23, only 2% of the genes codes for protein. Next, the repeated sequence make up very large portion, that is unique identification. Then the chromosome 1 has the highest number of genes, that is 2968. And if you see the Y chromosome, which is having very less genes, that is 231. So these are the salient features of Human Genome Project. In our next session, we are going to study about the advantages of Human Genome Project. Thank you.